nanohub.org.
Online simulation and more for nanotechnology.
I've been at Purdue for about 9 years now, and about 4 years ago, 3 or 4 years ago now, I started the Center for Implantable Devices, which is a teaming of various faculty in biomedical and in electrical engineering for the purpose of developing better, smaller, more capable implantable devices. Implantable devices to do things like, for example, detect seizures and stop them in epilepsy, to improve outcomes for patients in Parkinson's disease, to make it so they can move again. Implantable devices for neural control of robotic limbs for our injured warfighters coming back from Iraq and Afghanistan. Devices to treat alcoholism, depression, glaucoma, cardiology. There's a whole range of different diseases that can be treated by using electrical currents instead of pharmaceuticals. And so instead of pharmaceuticals, we call them electroceuticals, and that's the new buzzword. Yeah, so maybe a year ago, well, about a year and a half ago, I started working with Dr. Lundstrom. He put me on his grant for the NSF NEEDS, the NanoHub NEEDS initiative, Nano-Engineered Electronic Device Simulation. And the reason he brought me on is not because I am a guru in nano, far from it. I'm probably the least nano guy you'll talk to all summer, but rather because I'm on the end of applications. So folks who develop nano devices, like, for example, supercapacitors, and there we actually do some work, which is why supercapacitors is a supposed original title for this talk, which I've adapted. But then we end up working with guys like Dr. Tim Fisher with his graphitic pedals, making glucose sensors and turning those into a system. So if somebody makes a nano device, the question is, well, who cares, right? You can make a nanowire, right? Carbon nanotubes, nanoballs. You hear about all this stuff and you go, well, you probably don't, but I used to go, yeah, whatever, right? Who cares? What's the point? And at some point, you start to feel like people make things nano just because you can get funded if you use the word nano in your grant proposal. But really, why do you need nano? Like, why nano? And at one point, I started to realize that nano was no longer something that people did just for the sake of finding out how small they could go, but rather they were able to develop devices that you could not do if you did micro. And so nano was really enabling. And it enabled new applications. And I specifically am interested in implantable medical applications. And so I became part of the NEEDS team to try to find applications and integrate other people's nano discoveries into workable systems that can affect human health. And then last year we put together a preeminent team for the university, and we pitched it, and we've gotten a lot of support from the university. So we're not hiring faculty or just hired a full-time staff member. We're growing our space, our capabilities, and so on and so forth. Most recently, do you guys know Kyle Faxton and Chris Quinkert? They're both in my lab, and they're helping us right now with this concept of using supercapacitor-powered implantable neural recording devices to drive robotic limbs, and I'll tell you more about that over the course of the talk. But we have a deadline this Friday with DARPA, and that will determine whether or not we get an additional $13 million for the next 5 years. It's a lot of money. So that's why they're not here. Yes, and their scattered advisor forgot. And that's, so anyway, so that's their excuse. It's actually my fault. But anyway, so let me back up a little bit and let me talk a little bit about the needs in terms of the medical device space. And then what I'll do is I'll give you some examples of where nanotechnology enables specific important functions in our implantable medical devices. This all is more fun if you don't just zone out, if it's not that interesting to you, or if you have a question or whatever. Don't wait till the end. Just raise your hands and interrupt. It's informal, and it's always fun when you get interaction. So please feel free to just interrupt. All right, so what's driving all this? The medical marketplace is projected to reach almost 20% of the national economy by 2020, 19.5% by 2017. That's according to the Department of Health and Human Services. So 20%. That's 1 out of every $5 that's made in America will be spent on healthcare in some form or another. And yet, the practice of medicine hasn't really changed in the last 100 years, in the sense that individual doctors treat individual pathologies. So our goal is to enable multi-pathology treatment, treating multiple conditions simultaneously through physically distributed networks of nanoscale wireless sensors and actuators. So having multiple smart dust size, so tiny nano-enabled devices implanted and external and distributed throughout the body will allow us to track multiple conditions and treat multiple conditions simultaneously, treating the patient as a whole, rather than having your orthopedist look at your knee but not worry about your heart, which is your cardiologist, who's not worried about your eye, which is your ophthalmologist, who's not worried about your brain, which is neurologist, right? All these people treat things irrespective of one another. We're trying to do a holistic multi-pathology treatment. And this, we hope, will lead to a very specific outcome. And that outcome is a paradigm shift in the way that patients are treated. So in order to do this, we need to develop a number of technologies. These include technologies for sensing, for understanding what is currently going on in the body. Right? Most medical devices and most pharmacological treatments, believe it or not, have no sensing. They don't care what's going on in the body. So if you go to the doctor and you say, for example, you have a sinus infection, he gives you antibiotics. He gives you amoxicillin, 250 milligrams or 500 milligrams, 3 times a day for the next 10 days. While he's giving you these pills, while you're taking these pills at home, nobody's checking to see if your disease is responding, to see if it's responding more or less, to see if maybe, like most sinus infections, it's viral and not bacterial and you don't need to take antibiotics at all. There's no feedback mechanism. Right? If you have depression and you take antidepressants, nobody's checking the levels of your anti-inflammatory cytokines to see if you're actually responding to the treatment, or you're just, like so many, becoming addicted to Prozac with no therapeutic benefit whatsoever. So this sense that we need to know what's going on in the body while we're treating the body, this is actually, believe it or not, novel. And so we develop a series of different sensing technologies, and I'll give you a couple examples. Then we have to turn around and do actuation, which means we need to act on the body. We need to stimulate more of this or less of the other to get you healthy again. That's the actuating part. We need to network all these devices, which means we need to wirelessly power them, and we need to do wireless telemetry between devices. And then we need to have miniaturization. So we need to make everything small enough so that it can go into the tiny little nooks and crannies that are all over the body. And it would seem like nano is going to come into miniaturization, right? Because nano means small and miniature means small, and so you're going to see lots of nano here. Actually not. You're going to see lots of nano in sensing and actuating, and you're going to see some of it in powering, and very little in miniaturization, because as far as miniaturization is concerned, micro is good enough in the body. Okay, so let's talk about specific examples, and these are not meant to be comprehensive. They're meant to be examples. So one thing that we're interested in is glaucoma. Glaucoma is the second leading cause of blindness in the world behind cataracts. And it involves an excess of pressure inside of the eye, which leads to pressure on the inside of the eye. Excess of pressure in the anterior chamber leads to an excess of pressure in the interior chamber, which applies to the optic nerve, which kills the neurons. When the neurons die, they don't regenerate. Over time, you go blind. There are treatments for this. You can take drops, but what happens is that you give patients a drop, and, well, patients are all the same, right? They're the same as you are and the same as I am. A little warm, sorry, I just walked in from home 3 miles away. Anyway, so their patients are all the same. When you go to the dentist, how many of you go to the dentist? All right. How many of you go to the dentist twice a year? Fewer hands. All right. When you go to the dentist, how often does the dentist tell you to floss your teeth every day? Every time, right? Okay. Now, how many of you actually floss your teeth every time? Right, exactly. Everybody's the same, right? But what happens is that the week before you go to the dentist, you know you're going to go to the dentist, and you know he's going to ask you if you floss every time, so you start flossing. So when you get to the dentist, they go, do you floss every day? Well, you can say, well, yeah, sometimes. I try to, right? But the truth is, and you'll do it for about a week afterwards because you feel guilty because they just scraped your teeth clean and everything else and told you you have to floss. And by the way, they know you don't floss. They know you're lying because when you floss all the time, your gums get used to it. And when you don't, they don't. And so when they floss you in the dentist, they'll bleed if you don't floss all the time. There's nothing you can do about that. So they know right away that you're not telling the truth. But anyway, that's okay. We all lie. And it's fine. That's called patient compliance. And patient compliance is a huge issue in medical therapy. And just like you're better about brushing your teeth right before and right after you go to the dentist, but then you fall off, patients are better about doing things right before and after they see their doctors. So if you have glaucoma, you can't feel your glaucoma. You can't tell that it's there. And putting in drops several times a day is a pain in the neck, just like brushing your teeth or flossing them, right? So right after you see the doctor, they put the fear of God in you. They've told you that you're going to go blind if you don't do something about it. And they give you your drops and you take your drops. But after a while, frankly, you forget. And you stop taking your drops. And then when the doctor asks you, you say, of course I'm taking my drops, but you're not really taking your drops. And so when your pressure starts to go back up in your eye, the doctor doesn't know, is it because you're not taking your drops like most people, or is it because you're maybe not responding to the drops? In fact, they don't really know if your pressure is going back up at all, because the week you go, before you go see your ophthalmologist, you start taking your drops again. And so as far as they know, you're doing fine. But every time between visits, your pressure is spiking and you're losing vision. Over time, you go blind. What the doctors want is a device that's small enough that we can inject it inside of the eye, that will track the pressure inside of the eye and will tell them whether or not you're taking your medicine. Yeah?
How is that sensor powered?
That's a great question. We'll get to that with powering. It's wirelessly powered. So it's got to be small enough to be inside the eye, so we can't use a battery, so we have to wirelessly power it. So anyway, so what we did is this collaboration with Bill Chappell, who's a faculty member in electrical engineering, developed Danny Doyak Ha, who goes by Danny Ha, a graduate student who just graduated this spring, developed this pressure sensor. It's the smallest of its kind. It's small enough to go inside of the eye, and it allows us to track intraocular pressure. Now, it works, but it's very hard to build. It requires MEMS. It requires a lot of manufacturing expertise. They're flaky. You build 100, you get one that works. It's expensive. It falls gold. What could we do that might be better? So recently we've started working with a professor at MIT who has developed, Donna Weinstein is her name, who has developed something called a resonant body transistor. This is a nanoscale structure. And it involves nanoscale channels through which the currents flow in a transistor. And these can be suspended in air, and then they resonate. And they resonate at the frequency at which you want to create a resonator. So we can make a resonator and make it into a radio transmitter. And then that resonant frequency would be proportional to the size of the devices. But if they're suspended in air, the air has a certain pressure. And that pressure is going to have an effect on what the resonant frequency is. So could we take one of these nanoscale devices and make a better, smaller pressure sensor that we could integrate into a standard CMOS chip run? I think maybe yes. We haven't done it, but we're talking about doing it. But that's a potential application of nanoscale to a medical device. Taking something that's really cool like this, but it's still 500 microns and it requires a whole separate fabrication run, it's expensive, and making something nanoscale that we can integrate with the existing nanoscale CMOS fabrication run. Another one is neural recording. So if we want to be able to stimulate the brain to stop a seizure, we want to know when that seizure is going to happen. If we want to be able to stimulate the brain for Parkinson's disease, we want to know that the brain is responding to the stimulus the way that we would like it to respond to the stimulus. And in order to do both of those things, we have to be able to know what is going on in the brain currently. We have to be able to record. Excuse me. There's a problem. So whenever you get a splinter, right, the splinter sits in, like, have you ever gotten one of those deep splinters? The ones you can't pull out right away, but you just kind of see them under your skin. And you just basically just wait, right? And eventually your body kind of pushes it back out. Okay. Or dissolves it, or a little bit of both. All right. So when you put an electrode inside the brain, exactly the same thing happens. The brain sees a foreign object, and it doesn't like foreign objects, it doesn't recognize it, and there's an immune response. And what the immune response is that cells, called glial cells, begin to grow around the electrode to form a protective sheath to protect the brain. And that protective sheath has a high impedance, and it makes it so your electrode can no longer record small electrical signals in the brain. So you put an electrode in the brain, and it works really well at first, but over time it fails. And here's some photographs of commercially available electrode types, all of which 100% of the time fail. So in all of history, no one has ever been, you usually can't make sort of broad declarative statements like that. But anyway, in all of human history, no one has ever built an electrode that can record from a brain reliably for more than a couple of weeks. That's quite a thing to say, right? When we're trying to record individual neurons, not EEGs, that we can do. Or LFPs, we can even do that sometimes. But when you want to record single units, you put an electrode, and over a period of time, what happens is this thick scar, glial scar, forms around the electrode, and it makes the electrode fail. But it turns out that there's an alternative. And the guy who came up with this idea is David Yarrow. He's a former student in ag and bioengineering. If the electrodes are smaller than, say, 25 microns, the brain doesn't recognize them as a foreign body, and so it doesn't expel them. And so he had this idea, well, let's make them small. He's not the first to have that idea. The problem is that when you make them that small, they become really flexible. So when you push on them, it's like pushing on a rope, right? It just buckles. So he had this idea, instead of pushing, why don't we pull on them? Why don't we use a magnetic field to pull the electrodes down into the brain. And so that's exactly what he did. Here's a video demonstration in a jelly of a brain so that it's see-through, where we're trying to hit a 1 cubic millimeter target 5 centimeters down to the brain. It's shot at 20,000 frames per second, and the electrode goes down. It's like a bullet. We pull it right down. There's the target. And you can do it fairly repeatedly. So, well, there it is 3 times, but that's just the same video playing 3 times in a row. In fact, when we first made this video, we had to shoot it 12 times to hit the target, so it wasn't very accurate. We could get the depth, but not a lot of accuracy. We've since got some DARPA funding, and we're a lot more accurate now than we used to be. But this is a clever idea, and it allows us to do electrical recording. But what about recording other sorts of things? So, for example, Tim Fisher, Dr. Fisher, is he one of the speakers this summer?
Not this summer.
Okay. Well, he's one of the faculty members here in the building.
Yes, students in here.
Oh, yeah? Okay. Who here works with Dr. Fisher? Any of you guys, just you? Okay. Well, do you work anything to do with the graphitic pedals and the glucose sensing? Okay. You familiar with the glucose? Okay. Well, there's a glucose sensor that he's developed, and it's based on nanotechnology, on graphitic pedals that he grows here in Berk. These glucose sensors, well, he's got the sensor, but that's what he's got. That's it. What I have is I have everything else. I have the electronic. And it sounds like when I say I have everything else, like I bring 95% of the work to the table. It's actually the other way around. What I've got, we have, the world has. What he has is the really innovative part. It's the sensor. It's the interface with the body. It's the transducer that converts something we want to know in the body to an electrical signal that we can record with a microchip, right? And in this particular case, what he has is a glucose sensor. And so what if we could take a glucose sensor and we could put it on the tip of this electrode, which is microscale, and then embed it inside the body? What if we could do that, say, in a brain tumor? So I was having a meeting yesterday with a neurosurgeon, and we were talking about, she's a neuro-oncologist actually, so she works in cancer in the brain, and we were talking about how tumors work. And so what happens in tumors is that tumor cells metabolize differently than other cells. Regular cells use the mitochondrion to generate energy. Tumor cells metabolize glucose directly. So if you could have a glucose sensor that was in the tumor, you would know whether it's metabolizing more or metabolizing less. In other words, it's consuming more energy or less. If it's consuming more energy, bets are that it's growing. If it's consuming less, bets are that it's dying. And so if you're taking chemotherapy to address a brain tumor, and you have a glucose sensor embedded inside the tumor, inside the brain, you can track how well the chemotherapy is working in real time and tell the oncologist whether or not we should up the chemotherapy, drop the chemotherapy, or change the chemotherapy, right? That's kind of cool. So that's an application of nanotechnology to biosensing as well. And that's something that we could not, people like me who just make the whole device, could not possibly do without the nanotechnology. All right. Actuation. I'm going to go a little bit more quickly through this, but the idea with actuation is really just to reiterate what I was saying earlier, which is that the common practice in medicine right now is to stimulate open loop. So whether it's an electrical current or it's a pill, they give you the pill and they just wait to see what happens, rather than giving you the pill and checking immediately what's the body's response. There's a student a former student in my group, Matt Ward, who is now a research scientist here at Purdue, he had this brilliant idea, which seems obvious in retrospect, which is that if you want to control something neural in the brain, right, what you should do is when you stimulate, you should record the results of your stimulation. And then using signal processing, we can take the neural activity and we can break it up into neuron types. So in a nerve, for example, you have different types of fibers. So we can break it up into A, B, and C fibers, where A fibers are mostly motor neurons and C fibers are mostly sensory neurons. And then once we've broken it up, we can identify which types of neurons respond to which shapes of stimulus. And then we can adapt the stimulus so that it is responsive. So that you say, for example, you want to treat epilepsy and it turns out that a certain fiber type, C fibers, are involved with suppressing seizures, but A fibers are not. Well, we can stimulate C fibers and not A fibers. The reason that matters is you've all heard about side effects, right? So when you take a particular drug, it has the effect that you desire and then it has an effect that you don't desire. How many of you are over 21? All right, well, assuming those are the only ones here who've ever had a beer, if you had a few beers, maybe one beer too many, there's a desirable effect, which is you get a little dizzy and a little happy, and then there's the undesirable effect, which is you feel horrific in the morning, right? That's the side effect. No, it's a side effect. So the drug has something, it affects certain parts of your body, your brain in this case, what it metabolizes the alcohol, and it also affects other parts of your body because it has things that shouldn't have in it, like toxins, or because, well, the other parts of your body aren't really feeling like handling the alcohol anymore, right? So those things are side effects. You take a pill of any kind, it affects multiple regions. When you take a chemotherapy drug, the purpose of a chemotherapy drug is to kill cells. So what it does is it goes into your body and it kills cells. It's poison. Now, the hope is that it kills more cancer cells than it kills other type of cells. But if you've ever seen anybody on chemotherapy, you see that they're fighting for their lives, right? They're taking poison and they're trying to get it to kill the tumor before it kills the patient. And that's the balancing act, right? But what if you could deliver the chemotherapy drug only to the part of the body you want to deliver to, only to the tumor, right? That's what the drug delivery people work on. In electrical stimulation, that's what we're doing here. We're delivering the electrical stimulus only to the part of the brain or the nervous system that we want to stimulate, because we have tiny electrodes that we can put exactly where we want them to be. And then we're activating only the cells in that area that we want because we can do responsive stimulation. And I'll skip through some of this, but there's lots and lots and lots of applications. We're exploring many of them. Another tool that we've come up with lately is Optogenetics. And this is nanotechnology, but of a biological kind. In Optogenetics, what you're doing is you're engineering a virus. And what the virus does, what viruses in general do, is they go into a cell. They change the cell so that instead of making more of whatever proteins it usually makes, it'll make more of the virus. And then the virus multiplies. It doesn't do anything else. It doesn't serve any useful purpose. It just does that, right? Okay. So what if we made our own virus? So that instead of making more of the virus, you made a particular protein that you're interested in. For example, you could make a protein that you find in algae called a channel rhodopsin that is light sensitive, that opens and closes and creates an electrically conductive channel in a cell membrane. So that's what Optogenetics is. And it was developed by two guys, one at MIT, one at Stanford. The one at MIT is Ed Boyden. The one at Stanford is Carl Diceroth. They were working together, and they came up with this technique. And the reason it's useful is because it makes your brain light sensitive. And so now we can use photonics to stimulate different areas of the brain. And you can develop optrodes to deliver light of a particular frequency right where you want it to be. So for example, you could inject a virus that does half the work, make the protein in one part of the brain, and a virus that does the other half of the work, make the enzyme that splices the protein into the cell membrane in the other half of the brain. And then only the cells that go from A to B would get both viruses, and only those cells would become light sensitive. So if you want to know what the neurons that go from here to here do, you can use this technique, and then you can use light to activate those neurons only and nothing else. And that allows you to understand whether or not it's this projection or that projection or this connection or that connection that is involved with, say, your depression, or with, say, your epilepsy, or your traumatic brain injury, or any number of different things. And then once we know that, we can use the electrical device that I showed you previously to cure you. Here's an example of us using those viruses in a mouse. And so here's a mouse, and he's happy, he's minding his own business. And we put an optogenetic stimulator inside of him. You see the light goes on, that means we're stimulating. And when we stimulate, he starts moving around in left-hand circles. It's not because he wants to move in left-hand circles, it's because we're overriding his motor cortex and making him turn in left-hand circles. So we can control his motor behavior with light. This is pretty new stuff. Here he is again, running around, minding his own business, having a good day, and here we are ruining his day as he goes around in left-hand circles wondering, why the heck am I turning? I don't want to turn. Anyway, so before you have sinister ideas about us doing mind control and taking over your body, that's not what this is about. What this is about demonstrating that we can have a behavioral control using electrical stimulation. And if we can do it for something visible, it has to be visible, otherwise it doesn't make a good video, right? So if we can do it for something, no, I'm serious, that's why we do it visibly. We may do something visible. If we can do it that way, we can do it for something else. We can do it for example, seizure suppression. Epilepsy, incidentally, affects 1 out of every 100 people in the world. It kills more people in the United States than breast cancer. You never hear about it, do you? It's 1% of the population. And yeah, it's particularly prevalent amongst our injured warfighters. Right? These kids coming back from Iraq and Afghanistan, you hear about these improvised explosive devices. Those bombs used to kill people. And they still kill people, but less than they used to because body armor has gotten so much better. But what the body armor doesn't stop is the impact from hitting your head and your brain from bouncing around inside your skull. That causes traumatic brain injury, which has gone up dramatically over any previous war. Again, because they just didn't use to survive. But the traumatic brain injury leads to, among other things, epilepsy. Okay, so that's the actuating. You asked about powering. So that's involved with the networking. You're going to have more than one device. How are you going to network them together? Well, you're going to power them, and you're going to communicate with them wirelessly. So for the powering, we had to go back to nanoelectronics again. So with the powering, not nanoelectronics, sorry, nanotechnology. Yes, also nanoelectronics, because we make microchips for everything, but I'll get to that. So with the powering, the idea is that if you take something and you put a battery on it, the battery will be so big that it won't fit everywhere in the body. So if you want to put something inside of your eyeball, for example, you can't put a big Duracell in there. For 2 reasons. 1, because it's big, and 2, because eventually batteries run out, and who wants to have surgery again so they can get the battery changed, right? I mean, if it's just underneath the skin, it's a pacemaker, and you got to do it every 5 years, that's maybe not such a big deal. But if it's inside your brain, and you have to have brain surgery every couple of years to have the battery changed, it's not so good. All right. Believe it or not, I mean, we joke about these things, but that's the current standard of care. If you have Parkinson's disease and you get a device implanted in your brain, currently it's battery powered, and they got to change the battery every few years. All right, so we thought that was unacceptable, and so we said, well, first of all, let's do it wirelessly. So we can harvest energy wirelessly. There's electromagnetic energy in the air, Wi-Fi signals. We can make our own Wi-Fi signals and whatnot. You can take those, you can take an antenna, the antenna picks up the signal. That has some energy to it. If you optimize it, it has a slightly less tiny, but still tiny amount of energy to it. We can take that tiny amount of energy, and then we can store it locally, and then use that to power our device. But how do we do that? Local storage. Well, let's skip through a lot of that stuff. Oh, that's the demo. All right, well, maybe I won't skip through so much. All right, so how do we do the local storage? With supercapacitors. I'll come back to that. Other ways of powering involve inductive powering, strong magnetic resonance coupling. We've developed a technique, again, Dr. Chappell led the way on this, we call resonantly coupled filter energy transfer. So there's a lot of electromagnetics involved. How many of you are double E's? The rest of you what, MEs? What's that? You BMEs, really? Well, I know mine are BME, but okay. So we have a number. Raise your hand if you're BME. Oh, just one. Oh, okay. I was so excited. I'm still excited. That's great. And with my 2, that's 3. All right. So all right. So for the EEs, when you take electromagnetics, you'll start to learn a little bit more about this stuff. If you take advanced, electromagnetics and antenna theory and so on, then you'll really get into this stuff. Let me know if you want to. It's hard to find students who want to do electromagnetics. But we do spend a lot of time doing the antenna design and doing the rectifier design to convert the received wireless signal into DC electricity like a battery. And we get it to work pretty well. So here's a demo of a number of devices. Here's a device for intraocular pressure sensing inside of a rabbit's eye. Oh, no, sorry, this is the Optogenetics device. This is that is the intraocular pressure sensor. This is the cardiovascular pressure sensor. It's a little bigger, so it can go a little further away. That's our targeted muscle reinnervation sensor. I'll tell you more about that in a little bit. And if we make the devices big enough, we can power them 25 feet away. So this is all capturing energy out of the air and converting it to electricity to power a device. The next piece is the telemetry. How do we get the data in and out? For that, we do antennas. I'm not going to bore you with this. There's no nano. But we come up with some clever ideas for how to do the microchips that drive the antennas. We implement those on chips, which are themselves nano. But then the power storage. All right, so we don't have batteries. We don't have batteries because they need to be changed, because they have dangerous chemicals, because they're big. But we want to have some sort of energy storage, because if you're wirelessly powering something, it's not going to be very robust by definition almost, right? You always get dropouts. Your cell phone, you think about it, you get 3 bars and then 2 and then 1 and then no service and then back to 2 and then 3, right? But if it's a life-sustaining device, no service is a problem, right? So if it's a life-sustaining device and you go through a neuro service area, your life is no longer being sustained. So we have to have something to cover those areas, right, to provide safety. And really with medical devices, the first order of business is always making sure that you have safety and you have robustness and so on. So we came up with this idea of using supercapacitors. Actually, it was a student in my lab, Oren Gall, who had this idea, and he started developing the idea, and then he found this fabulous postdoc named Cuizhou Meng, who's still in my lab. Oren has graduated and gone on to Penn State, but Cuizhou is still there, and they started developing these supercapacitors together, which initially involved carbon nanotubes in a polyaniline matrix, pushed so close together that, your capacitance is equal to epsilon A over D, where epsilon is the relative permittivity, A is the area of the capacitor, D is the separation between the plates. So you move them really close together, you get a small d big capacitor, but really it's all about getting that A, that area, huge. And how do you make a huge area? Well, because instead of having a flat plate, you start putting curves into it. And that is what you do with nanostructures. Partnering up with Professor Tim Fisher again, they started using graphitic petals, and they were able to make the supercapacitors even better. Anyway, the end result is that they could make a supercapacitor that was a little less than 1 by 1 millimeter that would contain 1 millifarad, right, of capacitance. 1 millifarad, when I was an undergraduate student, would have taken a capacitor the size of this table. Right? So going from this to a square millimeter with a thickness of 20 microns, that's pretty cool. And that's nanotechnology. That's, by the way, what the whole talk was going to be on, but I don't really know enough about polyaniline matrices of carbon nanotubes to talk for a whole hour. So that's the problem with generally with being in my world. So in the biomedical engineering world, you're trying to put together work from a number of different fields. You're trying to combine electromagnetics, circuit design, nanoelectronics, all of the biology, the neuroscience, the medical knowledge, packaging. All of this stuff has to come together to make a full system, which means you don't get to be an expert in any one of these areas. On the flip side, if you work on a specific area, you focus, you can be the expert in that area, but then you don't get to build a full system. So there's sort of a trade-off. And at some point, you all get to make a decision in your careers, whether you want to go down one path or the other. Do you want to hyper-specialize, or do you want to be a generalist? And when you go to graduate school, you begin to make that decision. So I went to graduate school. I got a master's degree in electromagnetics before getting a PhD in neural engineering, but really I was doing circuit design the entire time. And then coming to Purdue, I said, I'm not going to do any animal work. I don't want to do animal work. I just want to focus on making my chips. But then nobody was doing animal work. I needed somebody to do it, so I started doing it. And now we run the biggest animal lab on campus. So you just sort of wander around and learn more and less, and it's all kind of fun. But I'm assuming you're all going to graduate school, right? Right, because at the end of the SURF program, you actually have to go to graduate school. Otherwise, they take you outside and shoot you. I don't think I'm allowed to say that. It's supposed to be a surprise. Anyway, no, they don't do that. That doesn't happen. But you should all go to graduate school, right? Because if you like this stuff and you want to be the one who's developing the technology, you need a graduate degree. If you like this stuff and you want to read about it in your magazine, then you can get a bachelor's degree. And now I'm not entirely kidding anymore, right? So the people who get to push the borders, who get to develop the new technologies, who really drive the field forward, those are people with graduate degrees. Undergraduate degree means that you work on the team led by a person with a graduate degree, and you do whatever they think you need to do next. And that can be fun and it can be rewarding, but you're not the one having the ideas. They are. That's a broad generalization. It's not always true. It's almost always true. So, even when you hear about people like Steve Jobs and people say, oh, the guy was a genius. He was a genius, but he didn't develop a single one of the technologies that he uses. Not one, ever. Now, he was able to recognize other people developing cool technologies and bring them together into a cool product, but that's an entirely different skill set. All right, so that's my graduate school pitch. The last piece of it is, so now we have sensing, we have actuation, we figured out how to communicate with them wirelessly, we figured out how to power them, how do we bring it all together, how do we miniaturize that? And the heart of that is the microchip. So I saved it for last, but this is a lot of what we do. This is a single microchip tape out that we did recently. It's actually got 83 microchips on it, but we fabricated this on the X-Fab last year. And it's got chips for all sorts of different things. Like, for example, here's a chip for glaucoma. So there's a chip that allows you to sense pressure. It's 700 by 700 microns. The supercapacitor that I showed you earlier was 700 by 700 microns. And that's not a coincidence. So the idea is that supercapacitor sits directly on top of this chip and powers it. And so this chip has a inductor, which allows it to create a radio signal so it can communicate wirelessly with the outside world. And then it's connected to an antenna. And the antenna can be very, very small. And so the two things together will allow you to do pressure sensing inside of the eye. The pressure sensor I showed you at the beginning of the talk, if you'll remember, was 500 by 500 microns. That's not a coincidence either. That pressure sensor mounts on top of this chip. So if you have a sandwich with the pressure sensor, the supercapacitor, and the chip, all together you have something that's less than a cubic millimeter that'll allow you, it's literally a grain of sand, you can drop inside the eye and it'll measure pressure. All right, so that's the glaucoma work. Seizure monitoring, the chip is a little bit bigger. And here we're recording from 4 channels in this particular case and allowing us to detect and track seizures in patients. That's for epilepsy. We also have stimulating ones, but this one is not one of them. Congestive heart failure, here's something very similar to the glaucoma device for sensing pressure, but using a different technology to track pressure inside of the heart. Electromyography is about recording muscle activity. And I'll tell you why we want to do that in a little bit. And you put all these things together with the nanotechnology, sensors, and actuators that I've mentioned, and combine them with a wireless powering and telemetry, and you get back to the patient. And so the idea, again, is that a single patient is, over the course of its life, not going to have a single condition. They're going to have a number of conditions. Like, for example, glaucoma, which would require a device inside of their eye, epilepsy, which might require a device inside of their head, heart failure, requiring something in their heart, a missing limb, which would require this muscle activity sensing for robotic arm control. Again, I'll get to that. What we're trying to do is we're trying to combine nanotechnology with conventional CMOS chips and novel ways of doing telemetry and powering so that we can treat all of these conditions simultaneously in a single patient. You can have a single hub, your iPhone, for example, with an app on it that tells you everything that's going on with your body. Actually, we don't really want that because the worst thing in the world is a patient with all of their own information, right? Because then they're constantly calling up the doctor going, ah! So what you really want is a device where everything gets sent to your clinician and they can see everything that's going on inside your body and they'll let you know if there's something to worry about. All right, hypochondriacs are the bane of the medical profession. And so that is why we teamed up with NEEDS. NEEDS I mentioned a little bit earlier, Nano-Engineered Electronic Device Simulation, led by Professor Mark Lundstrom. And here's an example of a transistor that's been modified. This is Professor Alam's work to be a biosensor. So we're trying to explore that and add that to the stable of technologies that we can integrate with our devices. So we take the chip that I mentioned, we combine it with Professor Alam's biosensor, and we can make something new. I mentioned the resonant body transistors and my hopes for using those for a variety of applications, including, for example, pressure sensing. Those are nanostructure, the MIT stuff. We fabricated it. So we can make them. We fabricated them and they work. We can package all of these things together. There's a flexible package made on Parylene. Graduate student Jimin Meng, who just finished his PhD this summer, combining a supercapacitor on a flexible Parylene package with a microchip, the antenna, the electrodes, the Parylene substrates, the whole 9 yards. The electrodes will be coated with a nanotechnology to do sensing of, for example, a specific type of neurotransmitter. The microchip itself is nanotechnology. The supercapacitor is nanotechnology. And so this is really a combination of technologies to create a full system to achieve a particular end. This one records muscle activity. We can make them smaller. I've talked a lot about size. This is how small we can make them. That's small enough to fit inside of a mouse. Not just anywhere in a mouse, but inside the eye of a mouse. So here's that intraocular pressure sensor I was talking about. There's Danny's pressure sensor, and we can couple that with the chip and the supercapacitor. There's the antenna. It's 2 millimeters in diameter. Goes inside the eye. We can make them even smaller. So here's a penny. And on the back of a penny, you zoom in, is Lincoln sitting in his chair comfortably in his memorial. And there's a microchip. And there's a wire which serves simultaneously as an antenna and as a probe to track something in the body. So this is flexible. It's also extremely fragile, as you can imagine. And so we can actually use the magnetic insertion system I showed you earlier for the electrodes. We can use that for the entire system. So we can use that, which is essentially like a little gun, and we can shoot a couple dozen of these sensors into your body and create a wireless network of sensors and actuators. And you won't feel them. They're too small to feel. And the gun, by the way, now this is entirely unprofessional, but imagine you've developed a new technique which uses magnetic energy to propel a small, sharp object a certain distance, right? At some point, you're going to get silly with it, right? And so we've actually been able to show that these devices we can actually embed inside of a door from across the room. So it really is a gun. In fact, the technology originally came from railguns. It was developed to put satellites into space. So everything that was developed to put satellite into space wasn't really developed to put satellites into space, right? And I'm not being a conspiracy theorist here. All of those satellite programs, they're about putting nuclear bombs into space, except then they can come back down. So all of the space programs were about getting ICBMs to work. And railguns was a cheap way of launching bombs into the air. That didn't involve all the rockets and propellants and so on. Anyway, they didn't really work for that purpose, but they've worked for a number of other purposes, including maglev trains, for example, and in our case, for injecting medical devices. So anyway, all right. I know that did sound a little conspiracy theorist, didn't it? But we're not interested in bombs, but we're interested in making people better. So we're hiring a bunch of faculty. I can skip this part, but let me get to this. Let me pause here for a second. So, all right. So this is Claudia. Claudia's a Marine, and so this is sort of the why we do what we do. Claudia is a Marine. She lost her arm in a motorcycle accident, I believe, but that doesn't matter. She lost her arm. And so the question is, can we give her back her arm? So they did a poll of Marines who'd lost limbs in combat recently. And in that poll, they asked them, what do you want, right? If we're going to build you an arm, we're going to build you a leg to replace the leg you've lost or the arm you've lost. What is it that you want back? You want the ability to pick up your kids, to play ball, to write, to scratch your nose. Imagine you've lost both your arms, right? No, I'm not kidding, right? Hey, all right, so you do a poll of quadriplegics. So let me back up. So long time ago, people did a poll of quadriplegics, folks in wheelchairs, paraplegics and quadriplegics. And they asked them, what do you want, right? And you can answer anything. So let's say paraplegics. What do you want? And you'd think the first thing would be they want to get up out of their damn wheelchair and walk, right? Absolutely not. It's like 10th on the list. Top of the list, always, they want to go to the bathroom. Right? It's not so elegant and it doesn't make such a great headline, but that's what they really want. They want to go to the bathroom when they want to, and they want to not go to the bathroom when they don't want to. Right? They want to be able to have sex and have children. It's a heck of a lot more important than walking. Right? So when you start asking patients what they want, you start to learn that actually what they want is not always the obvious. Right? I might say sex that the people in the room giggle, but it's a fact of life, right? So, and urinary incontinence and being able to poop when you want makes people giggle too. It's kind of funny. No matter how old we get, we still giggle about poop. Anyway, that's another story. So these things are the things that really make quality of life. And when you ask the patients what they want, they tell you what they miss the most. So back up to my poll with Marines. They asked Marines, what is it that you miss the most? What is it that you really, really want? And the top answer for the vast majority of the Marines polled was that they want to pick up their gun and go back into the fight. Surprising, isn't it? So what they want is they want to go back into combat. They define themselves as Marines, and their ability to fight for their nation is part of how they see themselves in this world. And what they've lost is that ability to fight for their nation. And so our task from our sponsors, the Defense Advanced Research Projects Agency, DARPA is a part of the Department of Defense. They're the guys who invented the internet, by the way. It's called the ARPANET before it was called the internet. Our task from DARPA was to restore enough motor control so that they can disassemble and reassemble an M16 in a couple of minutes, which is what they're required to be able to do to go back into the fight. That's a very tall order, and we're not there yet. But on the way to that, the task is that Claudia should be able to put on a robotic arm, and she should be able to control it with her mind. Now, when you put on a robotic arm, people initially think, well, the robotics, that's got to be really tricky. Yes, it is, but that's not the hardest part. We have very, very good robots. The hardest part is how do you connect the brain to the machine, How do you make it so that you can control that robot with your mind? You can make it so that you can control your robot with other motions, right? So that if you do this, shrug twice, your elbow bends, and you shrug once, and your elbow extends. No, that's how a lot of these prostheses work, right? With cables and whatnot, you make weird motions, and then your prosthesis does whatever it is you want it to do. And you can be trained, and it can work to some extent. And lots of people get by that way. But what we want is something more intuitive, something that doesn't require training, something that allows a patient to just go in and say, you know what, I want to pick up that water bottle. And when I pick up that water bottle, I don't think, all right, bend my elbow, move it forward, open my hand, close my hand, right? And I don't do things sequentially either, right? I do the whole motion all at once. I just will the water bottle to move up, and I don't know how it happens. It just happens, right? It's intuitive. Okay, you want the same kind of control. And so how do you do that? People started by saying, let's make lots of little sensors and put them inside the brain and record the brain's intention of movement, and then use that to control an arm. That sounds like a great idea. No one's ever gotten it to work, not realistically and reliably. And the reason is because it's very, very hard to record neural signals from the brain, as I mentioned earlier. So then people said, all right, well, the brain doesn't work, but the nerve is still there, so let's record from the nerve. Same problem with the nerve as you have with the brain. So people have had a little bit more progress with that, but it's still a very difficult problem. And then there's this guy up in Chicago named Todd Kiken, who's a doctor at the Rehabilitation Institute of Chicago, who had this brilliant idea, which is that you have a nerve that used to go down your arm, but the nerve stump is still there. And then you've got your pectoral muscle and you have your lateral muscles, but these muscles serve to move your arm forwards and up and down, right? You don't have an arm. So these muscles are now completely useless. What if you take the nerve that goes to those muscles and remove it? So you de-innervate the muscle. And then you take the nerve that used to go down the missing arm and you connect it to these muscles. It'll regenerate. And after a while, when you tell the patient, open your hand or close your hand, the muscle is going to contract in a particular way. And when you tell the patient, bend your elbow, flex your elbow, extend your elbow, pronate, supinate your hand, it's all going to contract in different ways. We can record the electrical activity in a muscle much more easily than we can in the brain. Because, first of all, it's a much bigger signal. And second of all, there's much less of an immune response. So the muscle essentially acts like a biological amplifier of the neural signal. So if we take those little devices that I mentioned earlier, these guys that we've made, and we put them inside of the muscle body, you'd be able to record her intention of movement. And right now, we can do this with the devices on the surface of the skin. And that's what the video is. So here's Claudia using her robotic arm the same way you use your biological arm to do a variety of different tasks. This doesn't require any weird twitching. It's just her using her arm and trying a variety of different tasks. Now, what you'll notice with each of these tasks is that each of them requires a different sort of movement, different hand grasp patterns, different combinations of elbow, wrist, hand, flexion, extension. Fine motor control. A different hand grasp pattern. This is going to be the hardest one of all. Oh, no, not this one. The next one's going to be the hardest one of all. Okay, no, not this one either. I haven't watched this video in a little while. This one. It's a saltine. And this is hard because it requires you to not just grab the cracker, but to not crush the cracker, right? And normally that requires motor or sensory feedback. And that's what we're working on now. I mentioned that I'm going up to Chicago on Friday. We have a big presentation and demo. We're trying to get a huge grant. Well, we've done this. Now we're going to try to add sensory feedback and combine the two things together. But these are the sorts of things that are possible when you combine a variety of different fields. Electromagnetics and electrical engineering, circuit design, package design, the mechanics of it, the biomedical understanding of it all. And then all of it is enabled really by the nanotechnology that does the powering, the sensing, and the actuating. Without that, you don't have anything. So that's what we do in the Center for Implantable Devices. Those are the sorts of technologies we combine, and that's what we do it for. Thanks. Yes?
I have a couple of questions. As I know, supercapacitors have a huge power density, but they give it all in a burst of time. They don't last, they don't store for many time. So when you manage the energy, when it goes up and down, how do you deal with that aspect?
Yeah, so what he's saying is there's two different things. There's power density and there's energy density. Energy density is how much juice can you hold. Power density is how quickly can you deliver that juice in a pinch. Capacitors have a lot of power density. Batteries have a lot of energy density. And so one thing is better at one thing and the other thing is better at the other. Supercapacitors actually sit somewhere between capacitors and batteries. They have more energy density, a lot more energy density than a regular capacitor does, not as much as a battery. They have a lot more power density than a battery does, not quite as much as a conventional capacitor. So it's sort of an in-between, best of both worlds kind of approach. Typically, when we make our systems, we actually combine a power supercapacitor with a regular capacitor, so we can have the same power density as a regular capacitor, which is good when you want to turn on quickly, transmit a burst of information, and turn off again. You want that power density. But then it has an energy density that is much closer to the battery, so it can act like a battery over longer periods of time. It's still not a battery, but at the same time, you can charge it and discharge it much more often than a battery. You don't need delicate charging and discharging circuitry. It's smaller, won't need to be replaced because the whole thing is wireless, and so on and so forth.
And have you heard of electricity use?
Of what?
Electricity, this is a term of wireless electricity. It uses reflections instead of these transmissions to power sensors in wireless. I heard of it.
That sounds like electricity sounds like a brand name. Do you know what the underlying technology is?
Okay, well,
Is it that sounds like the magnetic resonance coupling, but I'm not sure. How do you spell it?
WITR. Which is it? WIDRICITY.
This one?
I can't see anything.
Wireless energy transfer using resonant energy transfer. Yeah, okay. So this is based that. Yeah, this came out of the MIT work. This is magnetic resonance coupling. So one of the forms of powering that I talked about was, so in the far field you use an antenna to harvest energy. In the near field you use inductive powering. And inductive powering, typically what you do is you design a transmit coil that generates a magnetic field, and it resonates at that frequency, and you get another one that picks up the magnetic field, and it resonates at the same frequency, and then you put them near each other, and you get some energy coupling. If you put them really close together, it's almost 100%. That's how transformers work. But if you get any kind of misalignment, it drops off quickly. Magnetic resonance coupling, what it does is you take those two, and instead of designing them individually, what you do is you design them as a system so that they resonate with each other, rather than in each of them resonating independently. And when they resonate with each other, you get much higher energy efficiency, or coupling efficiency, over much greater distances. And you can take it a step further and treat the intervening space like a bandpass filter, which is what Chapel started doing, what we're doing now. And then you have this resonantly coupled filter energy transfer, where you get not just the advantages of magnetic resonance coupling, but you get a design matrix that allows you to compensate for motion, for tissues, for movements, for separations, for all sorts of different things, and do so dynamically. So yeah, that's in essence one of the types of powering that we're doing now. It's a great question. Yeah.
You mentioned your range for powering the devices was up to 25 feet in that video you were showing.
Yeah.
What's your range for telemetry and your information transfer?
That's the easy part. So the way to think of it is that when you're powering a device, you need to deliver power, right? So you need to have a certain amount of power. Let's say you need a milliwatt. So if you need a milliwatt to drive an implant, which is not unreasonable, then typically you measure power in dB with respect to a milliwatt. So 1 milliwatt is 0 dBm. So that's not zero power, that's 1 milliwatt. All right, so 0 dBm is what you need to power a device, right? To receive an electrical transmission and understand the data in it, you need minus 110 dBm if you're using an off-the-shelf receiver. So the amount of power that you need is 110 decibels less. It's huge, right? It's orders and orders and orders of magnitude. So it's much easier to do. So we don't really worry so much about the telemetry. We worry about the power efficiency. That's a good question, though. Other questions? That's it? Yeah.
I have a couple. They might be very simple, but for the pathways you're talking about in the brain, how you can simulate one part of the brain and then make it send signals to other parts of the brain. So what about things where there's multiple brain pathways?
Or what?
Multiple brain pathways.
Yeah.
So would that be a problem?
That's an excellent question.
Adapts and then they'll use another pathway.
Yeah. So there's two examples I can give you right off the bat. In Parkinson's disease, you have an inner and an outer loop for the feedback mechanism that goes through the basal ganglia. The substantia nigra dies, and what we do is, because it's dead, we can't go in there and stimulate it to make it work more because there's nothing there to work. So what we do is we find alternate pathways that project, that send signals to the same area, and then we stimulate those. And there's different ones that we can stimulate, and there's actually debate about whether you're better off putting a stimulator inside the subthalamic nucleus or you're better off putting a stimulator in the globus pallidus to get the same sort of therapeutic effect. And both are done in patients. In epilepsy, it's a little more interesting, I think, but maybe that's because I do most of my work in epilepsy. I don't know. But in epilepsy, I think it's a little more interesting because in epilepsy, you really have, it's a complex circuit problem. Sometimes you have a single focal area and you stimulate the focus and you can suppress the seizure. But more often than not, you have a number of little focal areas, and you have areas that project to and from different regions around the hippocampus, which is an area of your brain inside of the temporal lobe associated with learning and memory. And so the circuits, you don't want to be able to stimulate in one place because that's not really sufficient to prevent the seizure everywhere. And you also don't want to stimulate the whole brain, obviously, because then you'd be causing a seizure, essentially. So you really want to identify what are those areas of the brain that project to all of the different areas, or as many as you can get. And if there's one area that projects to many other areas, then it's better to stimulate that area, even though there's nothing wrong there, than where the seizure actually begins, because then you can affect multiple zones simultaneously. Does that make sense? Yeah. That's an excellent question. So understanding the circuitry inside of the brain is key to being able to deliver these therapies effectively. And our understanding of the circuitry inside of the brain is poor. That's generous. We have no idea, really. I mean, it's astonishing how little we know about how the brain works. Anything else?
I'm just curious, you said something about electroceuticals maybe being a replacement for pharmaceuticals, is that right?
That's what we hope to do.
Can you say a little bit more about that?
Yeah.
Like how that translation happens?
Well, I'm sure the pharmaceutical industry will have something to say about it. And I don't pretend that pharmaceuticals will be replaced everywhere. But I think there are areas, for example, in chemotherapy, we're not going to do electroceuticals, I don't think. I don't know, maybe someday. People do hypothermia, which is electromagnetic fields to ablate tumors. But anyway, there are specific areas in which pharmaceuticals fall short. For example, again, in epilepsy, because I work as much as I do in that field, 1/3, 30 to 40% of all patients don't respond at all to drugs. So for those patients, what do? Most of those patients have nothing. They can have surgery to have the part of the brain where the seizures start removed, but that's not optimal, right? That's every part of your brain is there for a reason. So nevertheless, some patients are desperate. Some patients have dozens of seizures every single day. That's not a quality of life that you want to have. And so in those cases, a brain resection is appropriate. But if we have a device that can go into the brain and stop the seizures, that would be nice. But even for the 2/3 of the patients that respond to drugs, the side effects can be horrific, right? You take a drug, you ingest it orally, it affects your whole body. It stops the seizures maybe, but it has lots of other effects. Nobody likes being on those kinds of medications. Neither do antidepressant patients or antipsychotic patients or any of those really strong drugs have terrible side effects. The promise of electroceuticals is that by identifying the area of the nervous system that you want to affect and by electrically controlling it, we'll do it without side effects, no side effects. So you'll be able to get the same efficacy as pharmaceuticals maybe, and that's good enough.
So isn't that the chemical stimulates something electrical, and now you're directly stimulating things electrically?
Exactly. So the way that two neurons talk to each other is that you have a neuron, has an electrical signal, travels down the neuron, and when it gets to the end of the neuron, it releases chemicals. Those chemicals act on the next neuron to generate an electrical signal. So there's always this back and forth between the electrical and the chemical domain, right? When you take a narcotic, for example, let's say a drug like cocaine, right? That's a simple one to understand. There's a neurotransmitter in your brain, which is called dopamine, which is actually what's involved in Parkinson's disease, a lack of dopamine in a specific area of the brain, the projections of the substantia nigra. When you take cocaine, the dopamine that is released in your brain, because neurotransmitters release it when they have an action potential, an electrical signal releases dopamine, and that acts on the next neuron to fire. It's an excitatory neurotransmitter. So you have that back and forth through the electrical and the chemical. That dopamine gets released. After a while, it gets reabsorbed by the cells. That's called reuptake. What cocaine does is it stops the reuptake. So the amounts of neurotransmitter dopamine that are released in your brain naturally are not reabsorbed as quickly, so they act for longer. And that's what gives you the side effects, which are the high that people enjoy and why they take cocaine again. The really fun part is that the brain will let you get away with that one time. But over a period of time, and it's pretty quick, it adapts and it says, when I release this much dopamine, it sticks around longer than I want it to. So I'm going to release less. And so it starts releasing less, which means now when you take cocaine, you don't get that high anymore. You now feel normal. If you want to get that high, you need to take even more. Okay? But the fun part is when you stop taking it. You stop taking cocaine, now the reuptake is working like gangbusters, and your brain's not producing enough dopamine because it's used to having too much. That's what they call withdrawal. Right? And it doesn't happen just with bad drugs like cocaine. It happens with medicines. It happens with all sorts of things. It happens with alcohol and alcoholics. It happens with nicotine and tobacco, but it also happens with morphine, painkillers, any opioid-based painkiller. It happens with all manner of things. Caffeine, right? How many of you drink coffee? Well, I can see you all drink Coke. How many of you drink, by the way, your dentist wouldn't be proud. How many of you drink coffee in the morning? All right. If you don't drink coffee, do you get a headache? Do you miss your coffee for a couple of days? Have you ever tried not taking any kind of caffeine for a couple of days? All right, try it. So caffeine increases the blood flow in the brain. When you don't take it for a couple of days, you get vasoconstriction, and it will give you a splitting migraine. And if it doesn't do that yet, you're lucky. You're young. It'll happen. Don't worry. So withdrawal mechanisms, it's all part of the same thing. So what we're trying to do with electroceuticals is we're trying to deliver the therapy without changing the chemistry and without affecting any more cells than we absolutely have to. And that way you'll minimize the side effects. Other questions? Okay. Well, thank you very much.